most important pillar of the HPLC interpretation, data analysis and interpretation is the interpretation of the chromatogram. So chromatogram is the graphical representation of the estimation of the compound or substance or drug substance by using the UV diode or FLD, fluorescent detector in HPLC. So after detecting through the UV or FLD, you will get a chromatogram like this. It is indicating that first from the recording, if it is a zero value and it's a positive value and it's negative value, the recording will be like on the baseline, because no compound will be detected till the baseline should be, there should not be any turbulence. It will be recorded like this way. After a particular time point, this is a timeline and this is a MAU. After a particular time point, for example, this is 1 minute, then 2 minutes, 3 minutes, 4 minutes, 5 minutes, 6 minutes, 8, 7 minutes run मैंने लगाया था. First the curve will be like the flat, and after some point of time, a small peak you can see. That most probably that peak is of the solvent. So that peak is called as ghost peak. That peak is called as ghost peak, and after this time point, it will go moving, and it will at 3.5 minutes. I got a peak like this. This a peak that is gradually uplifting, then it will be sharp, then gradually downlifting, and then it will be flat, and it is moving like this. Is it clear? So that peak is called this peak recorded is called a chromatogram. Is it clear?
Clear.
This peak recorded is called a chromatogram. So what are the components of this chromatogram? What we see in the chromatogram is, you can see an uplifting part, this one, and uplifting part, this one, and then it's a sharp curve will be there, and then a downlifting part, and then again the flat curve will be there throughout the run. Again flat curve will be there throughout the run. So what is the peculiarity of this curve? And you can see that this is corresponding. This corresponding area, you can see an MAU value. For example, it is 18 lakhs. Okay, 18 lakhs thing. So this is the estimated MAU of this chromatogram. So what are the components of this chromatogram? So if you see that this is a ghost peak, and this is a chromatogram, this is the height of the chromatogram, and this is the base of the chromatogram. And if you consider that this amount is 18 lakhs is a MAU dictated, but this height is almost 200 तक height है, तो ये 200 का 10% कितना है? 20 है ना? So here 0, here 20 है, तो इसका 20 तक हम एक लाइन ड्रॉ करेंगे. इसका 20 तक लाइन ड्रॉ करके, 20 तक लाइन ड्रॉ करके इसका क्वांटिटी टोटल ब्रॉड विड्थ we will calculate it as A plus B. Okay, what is A and what is B I will tell you, and you have to draw a straight line here, midline, like this. So this part is A and this part is B. So this is the fronting part and B is the tailing part. Front area and back area, tailing area. So A, how will you calculate? The most important peculiar feature of the chromatogram and this curve is this for each compound in specific experimental computation. This is the RT. This RT at 3.5 minutes will not change, should not change. It should be same. It should be repeatedly coming as 3.5 minutes only. It can come as 3.5 to 5.3 and 4.9 like this. Minor variation is admissible, but major variation should not come in a same experimental condition, same compound. It can change if the experimental condition is changed. And this MAU should be same for same compound, same experimental condition, unless until the concentration is changed. If concentration increase हो गया तो MAU increase होगा. Concentration decrease हो गया था. If the concentration is doubled, MAU will be like this. Okay, and if the concentration is halved, MAU will be like this. It become half. So that is there. So A is the fronting part, B is the tailing part. And what is tailing factor? tailing factor is an important parameter. We can ensure the accuracy of the chromatogram. If the tailing factor should be always less on 1.2k बीच में आना चाहिए. So how will you calculate the tailing factor? So you have to take a plus b width divided by 2 into a. This is the equation to calculate the tailing factor. A plus b divided by 2 into a. So if you got a value like 0.9, what is meaning of that? 0.9 tailing factor आ गया तो what is the meaning of that? Because if a 0.9 tailing factor आ गया तो this will be like this. Means tailing नहीं fronting है, front part of broad है. If a tailing factor आ गया 1.4, how will the chromatogram look like? This chromatogram is एकदम ठीक है, but it will come like this way. ऐसा होगा. अब समझा है ना? So that is not admissible. Ideally, tailing factor will be between 1 and 1.25. इसके बीच है तो we can take that chromatogram and we can interpret that chromatogram. Otherwise, you shouldn't take that chromatogram and interpret that chromatogram. It will be wrong. गलत होगा. So fronting factor and tailing factor is the most important factor while interpreting the chromatogram. RT is called a retention time. Always you take a 10% above, 5% or 10% above baseline से ही करना है. Why you are doing like that way? Why doing like that way? Because always there will be small turbulence in the baseline of the chromatogram due to some other unknown compounds or the base plasma के interpretation में baseline turbulence होगा और turbulence के ऊपर ही हमें measurement करना है so that it should not interfere with the measurement. इसलिए it is called a noise of the chromatogram. So here another terminology is there, sound noise ratio. And we will discuss later. Sound noise ratio is also important for the validation process. So third terminology is number of theoretical plate. Number of theoretical plate. So number of theoretical plate is a number of plane, three-dimensional plane inside the column by which the molecule is traveling. So higher the number of theoretical plate, better the efficiency of the column, better the detection accuracy. So in a new column, if you purchase a new column like C18 column, this theoretical plate will be always 12,000, 15,000. After a long run or 3-year run or 3,000 runs or 1,000 runs, this theoretical plate will come down to 5,000, then 3,000. Then 3,000 के नीचे तो it is cannot be taken. That column is being weaned off. We cannot use that column. So these the things are main things to be considered by chromatogram to be interpreted. If we get a chromatogram like this, whether it is good or wrong, this is another possible error. It is broad chromatogram. It is not so good. This is on error. This is due to column property or the compound property, adhesion to the compound, adsorption to the column, silica particle. This thing can occur. And another error can occur in the chromatogram is like this way. Notching of the chromatogram. This is also an error. Another one is like this way. This way. Take off from end. Or sometimes take off from beginning. Sometimes two chromatograms are there like this way, and this is a good one because it is easily this is called this and this chromatogram is resolved out, completely resolved out from each other. If this is like this way, it is not resolved out, it cannot be taken. So these are the error in chromatogram that you can see. One is this is resolved out. Initial one I have shown is resolved out, this one, because this is and this is acha pk sharp and resolved out. This is and merged together. So it is being merged out together. This chromatogram you shouldn't take. Sometimes a turbulence itna hoga. Whether you can take this chromatogram or not, you cannot take this chromatogram. Sound noise ratio high. In that scenario also you cannot take this chromatogram. So we given a minimal description about the chromatograph and what are the errors, possible errors happen in the chromatograph, and what are the sound tailing factor, what is mean by tailing factor, what is mean by retention time, what is mean by sound by noise ratio, and the number of theoretical plate equations we will discuss in next section. So clear?
Okay.
You can stop.